And we're good. Okay. So this cover slide is the same as the first one that was posted on Canvas from Monday. We're going to get into some different topics. The whole slides are named differently when you go and find them on Canvas. They are named comparative genomics. And yes, I know there's a typo in the title. I also discovered that genomics, I left off one of the letters in genomics in the title of the slides. Sorry. All right, so some key terms. First key term, single nucleotide polymorphism. And so this refers to places in the genome where there are multiple nucleotide options at a particular site. Okay? So it's a particular site. It could be in a gene, it could be in a coding gene, it could be in just any gene, or it could be in what we call intragenic regions where it's not an actively transcribed site in the genome, but it's a place where it's not conserved. As you look at different human genomes, the particular allele that sits in that position varies. Okay? Now, for a long time, single nucleotide polymorphism, that term was only used if greater than 5% of individuals have the variant. So if 96% of humans had a G in that position and 4% had an A, it would not be called a single nucleotide polymorphism. Instead, it would just be called a point mutation. Okay? And unless you went over that threshold of 5%. Recently, that condition has relaxed a little bit. Okay? And so now we'll use the term single nucleotide polymorphism regardless of how many individuals have that variant. And then we'll just use terms like common, what's the other one? So common is if it's over 30%, and then there's like moderate, I think is the other one, and rare. Rare if it's, under a particular percentage. Okay. Now, initially following the publication of the human genome, all of the differences among genomes were assumed to be these, single nucleotide polymorphisms. But lo and behold, we have, not yet. All right, I thought my next term I should have looked to see what's coming next. I got excited. The minor allele frequency is just whichever is the rarer of the two forms with a single nucleotide polymorphism. Okay? So if 70% of people have a G in that position and 30% have an A in that position, we would call the A the minor allele frequency. Sorry, it may get cold in here, but the heater's freaking the projector out. Yeah?
Can you repeat yourself? The rare of the two forms with what?
The minor allele frequency. That's what this term refers to. It refers to the rarer of the two forms at a particular site where you've got a single nucleotide polymorphism. Yeah. Now, lo and behold, we have what are called copy number variants. And this is where you actually have a chromosomal mutation. It's not a point mutation. It's not switching a purine for another purine or switching a purine for a pyrimidine. This is where you actually have a chromosomal mutation, a deletion, a duplication, an inversion. And so you've essentially changed the number of copies of that you have of a particular region on a chromosome. Okay? So now typically these copy number variants are going to be in the thousands of bases, so like in the kilobase size. But occasionally they're in the megabase size, in millions of bases. Okay? And most often these are duplications, but sometimes they're deletions. Yeah, David.
What's a quick definition for minor allele frequency?
The rarer of the two alleles for a single nucleotide polymorphism. Yeah, Sadie.
So of the two, of the variants of the single nucleotide polymorphism, it goes just that one and then the copy number variants and then minor frequency and
Well, if you have a single nucleotide polymorphism, you're always going to have a minor allele frequency. Right? So it's whichever's the less common, or if you've got multiple, like there are some sites where all 4 bases show up when you look at all human genomes, and then so you'd have the dominant allele, and then you'd have 3 different minor allele frequencies, 1 for each of the 3 other bases. But if you have a single nucleotide polymorphism, you're always going to have a minor allele frequency. We'll see this a little bit later, but there are millions of single nucleotide polymorphisms in the human genome. Okay? Now the total number of bases is basically however many single nucleotide polymorphisms, it's that number of bases. There are far fewer copy number variations, but some of these are again dealing with a single variant is several million bases in length. Okay, so the overall impact on the genome is larger here, even though this is a much rarer difference among individuals. Okay? But it's the amount of impact is much, much larger. Yeah, Chad.
What effects would be felt by having an extra copy of
Depends. Yep, that's an excellent question. It depends a lot on what portion of the chromosome is duplicated. If you've got a coding region that's duplicated, it depends on what protein that's coding for. If it has to be heavily controlled, how much protein product you make, it's going to be devastating. Okay? Even if it's in a coding region, but it's a protein that even if you have a lot of it, it's not going to be detrimental, then it can have very little impact. Yeah, that's a good question. Most of these copy number variants, they do not have a large impact on phenotype. Okay? Now some of them do, most of them do not, because they're in non-coding regions and they're in non-regulatory regions.
So the copy number ones don't affect phenotype?
I'm saying that's not a rule that they don't impact, but most of them have little impact, and that's why they persist.
So the single nucleotide polymorphism.
That's true for both of them. Most of them have very little impact on the phenotype.
What was the one you said that had a lot bigger effect?
Nothing today.
You said.
Now I said it had a larger. This one makes up a larger percentages of the differences among people, the genetic differences. Yeah. Does that clarify?
I had a different question.
Oh, okay.
You said the duplication was more common with the copy number?
Yes. Okay. Yeah. Okay. Any other questions? We're going to talk about both these terms in more detail. So if you're like, oh, I don't get it, we'll talk about them in more detail, and you may still feel that way, but then you'll have at least some more words to work with. All right. Neanderthals, Homo neanderthalensis, and so this is another species member of our genus that almost undoubtedly has interbred with our species. Okay, we have markers that don't mark to any other early versions of our species, but they mark very significantly with Neanderthal genomes. Okay, we'll talk more about that in a minute. Denisovans, individuals in the Denisova or Denisovian region of Europe, and this is a little bit hard to figure out what species this is, but it looks most likely that it is Homo erectus or something very similar to Homo erectus. And again, almost undoubtedly, our species, Homo sapiens, interbred with this species as well. Okay, we'll talk about that a little bit later and what that actually means. All right, the last key term, encode. So encode was basically a project similar to the Human Genome Project, but rather than being tasked with sequencing the human genome, it was tasked with figuring out what the human genome actually does. Okay, because after the Human Genome Project, we found out that basically about 1.2% of our genome is coding, is protein coding. And nobody really believed that 98.8% of the human genome was junk. Okay? And so they basically tasked a group of researchers to figure out what the other 98.8% does. Okay? And this group was the ENCODE project. And they published most of their findings in 2012. as a series of 30 papers spread among several journals. Okay, and we'll talk more about the ENCODE project in a little bit. Okay, any questions on these key terms?
What was the one for Neanderthals?
What's that? Neanderthals? Another species in our genus that has interbred with our species. Same thing with Denisovans.
Are we talking about differences between them or not?
What's that?
Are we going to talk about differences between them or just like that's all?
I mean, only to say that there are very clear genetic differences among these species. Like if I gave you an individual, you would have no problem figuring out genetically and morphologically what species it belonged to. Okay. All right. So some findings. We left off here on Friday, so I'm going to go boom, boom, boom. Okay. We went through all of these on Monday, not on Friday. Today's Wednesday. Gosh, what a wonderful reality that today is Wednesday. And next week is spring break. All right. So the last finding of the Human Genome Project that we're going to talk about is this one, that paralogous, orthologous, and pseudogenes are incredibly common in the human genome. That there are a number of examples of paralogous genes in the human genome where you have two very similar genes that are coding for very similar protein products, and they exist in what we call a gene family. Okay, I gave you an example on Monday of our hemoglobin genes. We've got a number of genes in the MHC family. We'll talk more about that today, that are responsible for our immune response and our detection of foreign material. We've got a number of examples of orthologous genes in the human genome, and that's where we have to make a similar protein product to what other animals do. Okay, and we can compare our version of that gene to their version, and if we share ancestry, then those would be orthologous genes. If we don't share ancestry, then I guess you could still call them orthologous genes, but the explanation for how they exist would be different. They don't exist because of shared ancestry, they exist because of shared design. And then pseudogenes. Pseudogenes are incredibly common in the human genome. I forget percentages, but it's an unusually high percentage of our genome that looks like at one point it was made up of transposable elements that were mobile and have now frozen inside of our genome. But there are still a few regions. Sorry that it's cold and windy, but we got to keep our projector happy. Now I got distracted. Oh yeah. And so there still are a few sections in the human genome that are still mobile. Okay? All right, so let's talk about pseudogenes, and this will allow you to answer question 2 on the quiz that was due almost, what would it be, about 38 hours ago? So pseudogenes. And so the explanation for pseudogenes, if you are operating under the assumption that all living organisms root back to a single living ancestor, then complexity has to increase through time. Okay? Genetic complexity increases through time, structural complexity increases through time, ecological complexity increases through time, right? Does that make sense? And so you get an explanation even out of our very own text. I don't know if you've read through chapter 8 of the textbook or not, but basically when it's talking about the genetic requirements for multicellularity and how incredibly complex it is to be multicellular at the genetic level, because you need to develop division of labor among your cells, that's basically, there's a sentence in the textbook that says this is why it took 3.5 billion years to develop the first multicellular life form. That life basically existed for the last 4 billion, just shy of 4 billion years, but multicellular life didn't show up until a little bit over 500 million years ago. The reason why it took 3.5 billion years is because it's really complicated genetically. It's really complicated genetically. Okay? So operating under that assumption that genomes and morphologies and ecologies are going to get more complex through time, you basically, if you find 2 sections in the genome and their sequences are very similar, the best explanation for that's going to be, well, it started as 1 copy and now it's multiple copies, right? Because one copy of it would be simpler than having multiple copies, right? If you're going to start talking about multiple copies being there from the beginning, then you need some mechanism by which you can generate new material in multiple forms simultaneously. It's like, wow, that's not going to work. But there's some beauty to that, but remember that, okay? All right, so you see these sections in the human genome where you're like, man, there's a section on this chromosome that it looks very similar to a coding gene on a different chromosome. The difference is it doesn't have the promoter region, okay? It doesn't have one of several promoter regions. There are 70,000 or so promoter regions in the human genome and 20,000 or so genes, 21,000, okay? So oftentimes genes have multiple promoter regions. Anyways, but it looks very similar. And so you're like, wow, it's so obvious that the origins of this were something like this. We've got this mechanism by which sections of the genome become mobile. And so here's what happens. Here's what happened, okay? This section, this coding gene gets transcribed, okay? It gets transcribed into RNA. And then we have a machine called reverse transcriptase that takes that RNA, writes a DNA version of it, and then you have another machine that will insert sections of DNA that are free-floating that shouldn't be like that into the genome somewhere. Okay? And so we're like, all right, well, this is what happened. Basically, you had this original gene. It was being transcribed like usual, right? It gets transcribed into RNA. That RNA matures into mRNA. That mRNA gets translated into protein. But some of them some of them go through reverse transcription and get written back into DNA. Okay, and then from there into double-stranded DNA, and then from there being put into the chromosome. And you're like, what a wonderful story. Because that explains how we can get two copies of the same thing. And why you would lose the promoter. Because the promoter is not part of this RNA transcript. Right? Right? Just the actual part of by the time it matures into this mRNA, the introns have been cut out, the promoter wasn't there, and the 5' and 3' ends have been cut down a little bit. And so now you're going to have something, when it's written back into DNA, it's going to look a lot like the exons of that gene, but that's it. Promoter's gone, introns are all gone, it's just the exon sitting there. And you're like, this makes so much sense. And then on top of that, we find there are still pieces of the human genome that still do this. That they get transcribed into a immature version of mRNA. They mature, they have the introns cut out, they process it just like you would to get the mRNA to leave the nucleus and be translated. But then the machine reverse transcriptase takes them and writes them back into DNA. And it provides what we call copy and paste translocation, or copy and paste transposable elements. And you're like, there's still portions of the human genome that do this, and so it makes sense how we can develop these pseudogenes. Yeah, Sadie.
Is it just like a random process, or are there specific...
No, it's not a random process. There are specific regions that have to have a specific sequence on either end, at the beginning and the end, that are called the insertion sequences, that signal the machine to know that this is a transposable element and needs to be dealt with in this way. Yeah, it's a good question.
So what criteria would a piece of genetic code have to meet in order to qualify to be that?
Well, if it was under proper design, it would need to have a specific sequence at both the beginning and the end of this. And so that it basically signaled to the machine that it signaled to reverse transcriptase that it needs to be written back into DNA and then inserted somewhere else.
So the codon that that sequence codes for is for it to be...
No, it's not a protein. It doesn't get translated. So the codons are irrelevant. It's just the beginning and the end of the sequence match a section somewhere else where it can be inserted efficiently. Yeah. Yeah.
So does this happen in stem cells or in regular?
It does. Well, both, but it happens primarily during early development. and is a major mechanism by which the cells change a substantial amount in development, right? Because if you move some of these transposable elements into various parts of the genome, it can shut off genes permanently by basically putting something right in the middle of the gene.
So like, for like a skin cell, if you look at the genome of a skin cell, it could look different than like a liver cell because of this.
Oh yes, absolutely.
Like the,
yeah, and so we say that all of the cells in our body are genetically identical, but if you actually if you sequence the genome from one of your skin cells, not at the very end because they're enucleated, but if you sequence one from like a stem cell in your deepest layers of your epidermis and the dermis, and you sequence that genome and compared it to the sequence of a genome from a liver cell, they would be different, undoubtedly, and sometimes in very big ways. Yeah, sometimes in very big ways. All right. Now the question is why? But what's really interesting, and here let's talk about this. These pseudogenes become the bulk of the regulatory elements of coding genes. Okay? And so here's how that works. One is these pseudogenes, if they still have the promoter, right? So if the promoter, or if there's a promoter upstream of them, it may not have come this way, but some of these pseudogenes still have a promoter, and we call those duplicated pseudogenes. They can be overexpressed. They can be expressed more highly than the normal mRNA, and they can compete with that mRNA for translation. But then when they get translated, it doesn't really build anything normal. They're competing with ribosomes. They're competing with the normal mRNA for ribosomes, and they lower the level of translation of the normal mRNA. Yeah, Jack.
So where it was reinserted, it already had a promoter.
Correct.
The promoter's not getting reinserted.
Yeah, the promoter's not going to get transcribed, so it would have to be inserted into a region where there's already a promoter region. Yeah, it's a good question. Okay, so this is one of the more minor or rare ways in which these regulate the expression of the parent gene.
Wait, so you said pseudogenes become the most regulatory?
Yes. The bulk of the regulatory elements of other genes. Yep. So they can be expressed as what we call site inhibitory RNA or single inhibition RNA or small inhibiting RNA. And what these do is these go and in the cytosol they bind to mRNA that they're complementary to. And if you bind to mRNA, tRNA can't read it. The ribosome can't read it. It can't be translated. Okay, so these small interfering or single interfering RNAs, basically, if this, again, gets put into a place where it's promoter, it's going to get written into RNA, and it's going to have a sequence very similar to its parent gene, and it can be complementary to that mRNA, bind to it, and prevent that mRNA from being translated. Okay? And these are some of those cytoplasmic elements you inherit from mom, are these small inhibiting RNAs. Okay? That regulate the expression of something else. So let's say, your genomic imprinting from your dad is saying make a lot of this proto-oncogene. But you've got all these small inhibitory RNAs in your cytoplasm that you got from mom that even if you're transcribing a lot of that, they go and bind to it because it's complementary, it doesn't get translated. You're like, that's so beautiful. Thanks, Mom.
Would that be kind of a waste of energy though?
Yes, unless you look at it as the male and the female are competing, right? And so it's not really a waste of time, it's who's going to win. And competitions are never a waste of time. Okay. Yeah, this is similar to these small interfering RNAs, but it can bind to them while they're still inside of the nucleus, as what we call antisense transcripts. And then we have another type of regulatory RNA called microRNAs that basically regulate the rate of transcription. And so if you've got this pseudogene that looks a lot like this parent gene, it can compete with that parent gene for these microRNAs that are going to increase the rate of transcription. But they're binding here, and increasing the rate of transcription that's never going to produce a protein product. Right? Right? So preventing the microRNAs from binding here, increasing the transcription of something that will produce a protein product. Okay? And so these pseudogenes function in a really wonderful way to regulate the expression of the similar gene. And again, maybe even the bulk of the regulatory elements are in this format. And so you're like, now this is really interesting because I can see how we might get a pseudogene in the first place, but the proper amount of transcription of the true gene depends on the presence of the pseudogene. So either it's been around long enough that the machines have learned and have come to depend on it, right? It's like when you drink coffee enough, you come to depend on it. I don't know if you know this or not, but you can live a perfectly fulfilled, gratifying life never taking a sip of coffee. I know, it's hard to believe, but it's true. So either they became dependent upon this because it's such a wonderful design, or that this is the way it always was. That for every gene, for every coding gene in the genome, you're going to have at least one pseudogene as necessary to regulate its expression. And so now we've got, and that's probably the case, and so we still have, even though we have a mechanism for creating these pseudogenes, they still have to be there from the very beginning, otherwise you can't properly regulate the expression of that gene. Yeah, Chad.
Does that mean you inherit some from your parents and also make some of your own?
Yeah, now there are very few parts of the human genome that are still active as transposable elements. There are some, there are few, but it looks like in our history there were far more. And we could talk maybe about why that's the case and why there's a kind of a trend in transposable elements in all organisms towards becoming rarer and just fixing and ending that. But anyways, that's another story for another time.
Wait, but what would be the purpose of fixing it? Because each cell is different. So isn't the whole point of it?
Well, because it can be devastating, right? So if you move this transposable element into a gene, it's going to destroy that gene, right? So there are several genetic illnesses that correlate very significantly with overactive transposable materials, transposable elements. Because everybody's not equal. Like your genome might be more active than mine as far as transposable elements are concerned. Okay? Does it make sense? A little bit? Okay. And so pseudogenes, the question 2 from the quiz that was due like 38 hours ago or so. I don't even know. I don't really want it. Wait, so it's 36 and some change. 36 hours and some change ago. The answer is pseudogenes. Of all of them, there are going to be issues there or implications there are going to have the most heavy influence on the regulation of genes. All right. So now on to the rest of Chapter 8. The rest of Chapter 8 is all about comparative genomics. Okay, and so it begins basically with an explanation of, after we finish the Human Genome Project, and we've been accumulating now thousands of different human genomes, how do we actually differ? How different am I from you? How different are you from your, somewhat distant relative? And how different are we from humans that were alive, several thousand years ago? Okay? And so how does this all work? And you see there are 2 different areas that we're going to talk about. What are these? Single nucleotide polymorphisms. And what about these? Copy number variants. Those are 2 common ways in which we differ. Okay. There are about 54 million single nucleotide polymorphisms in the human genome. There are about. And the reason why I say there are about is because I may have slightly over 54 million, you might have slightly under 54 million, but it's right around 54 million. And you're like, that's pretty cool. And so based on that number, and this is what you started to hear immediately after the Human Genome Project was concluded in 2002, is that we are 99.9% identical at the level of the genome. If you're looking at just these single nucleotide polymorphisms, okay? 99.9% identical at the genome level. And people freaked out. They're like, one, super cool, we're so similar. But two, how can we be so similar and yet so different? And we know now because epigenetics is just a wonderful thing. It's just a wonderful thing. But anyways, that's another story for another day. Not today. Okay, so this is basically, of these 54 million single nucleotide polymorphisms, less than 0.1% of them change the primary sequence of a protein. Okay? So of these 54 million single nucleotide polymorphisms, less than 0.1% of them change a single amino acid in a single protein. Okay? So like, that makes sense, right? If we're going to maintain this variation, it's probably not going to have a big impact on the proteins that have to be made. So very few of these are in exons of genes. Most of them are in introns that get cut out as you're processing immature mRNA. And then a lot of them are in what we call intragenic regions, in areas that don't get transcribed at all. Oh, that's right here. Most are found in introns, followed by intragenic regions. Very few of them are actually in exons. Now, most of these, this isn't saying most correlate with disease. This is saying most that correlate with particular genetic illnesses. So there are particular genetic illnesses that are the result of a single nucleotide polymorphism. Great example, sickle cell anemia. Okay, it is a single nucleotide polymorphism and it results in genetic illness. Okay, another great example, many of the mutations in the CFTR gene that result in cystic fibrosis are single nucleotide polymorphisms. Not all of them, there are over 100 different mutations, but so most that correlate with disease are not in coding regions. Most of them that correlate with genetic illnesses are single nucleotide polymorphism in regulatory genes, like pseudogenes. Or genes for transcription factors. Or genes for DNA repair mechanisms. Okay?
That makes sense because most of the genome is not.
Yes, most of the genome is not coding.
So that's what we
Yep.
expect.
Yeah, you're absolutely right. Okay? So that's single nucleotide polymorphisms. If all you consider is those, we are 99.9% identical genetically. And you're like, that doesn't make any sense. I thought I was only half similar to my mom and half similar to my dad, but yeah, that's what we're talking about in the variable regions. You know, half of your variable markers came from mom, half of your variable markers came from dad. But anyways, but we were like, okay, well that's kind of cool. But then we start to realize that these copy number variants are really common. In fact, about 12% of the human genome, so the human genome is just over 3 billion bases, so 12% would be just over 35 million bases, I believe, if my numbers are right, are impacted by these copy number variants. Is that number right? 10% of 3 billion would be 30 million. Yeah, no, 10% would be 300 million. Oh my goodness, I was not right. Okay, so we're looking at like 350 million bases impacted by these copy number variants. It's a huge portion of the genome. These copy number variants influence about 13% of the coding genes. So of the 21,000 or so coding genes, about 13% of them are impacted by copy number variants. Either a duplication, or a deletion, or an inversion. These big chromosomal mutations.
24,000 coding genes?
21,000 coding genes. 13% of them impacted by copy number variants. So what this does is it pushes the differences among humans as high as 3%. Now, for most individuals, we're about 0.5% different. So for most humans, we are 99.5% identical at the level of the genome. So where you're like, well, where's this number 3%? These are incredibly reproductively isolated human groups. Okay? Incredibly reproductively isolated human groups. Like Aboriginal peoples in Oceania. Okay, very highly reproductively isolated tribes in South America and Africa. Okay, that you can get as high as a 3% difference at the level of the genome. Okay, that number is going to be interesting when you see other numbers that are presented in comparative genomics. Copy number variance makes it to where even identical twins are not identical at the level of the genome. Because most of these copy number variants happen during development. Most of these copy number variants spring up during development. And so even identical twins are not identical at the level of the genome, which probably should make sense because there are subtle differences in identical twins. Okay, and it's not just environment, it's also what's in the program. Yeah.
When the first clone sheep had like a spot on it that's more different.
Oh, I don't know. I don't know much about Dolly, except for that she died really young, and she was huge compared to, you know, a normal sheep. All right. So we have a region of the human genome. Actually, we have several regions. It's a whole gene family called MHC, and that stands for, if you're curious, you don't need to know this, Major Histocompatibility Complex. Okay? But it's a section of the genome that is really important for you to be able to detect self from non-self. So it's something that they check for when they're looking for organ donors, to make sure or to try to eliminate the opportunity for a recipient to reject an organ. This, of all the sections in the human genome, this is by far the most variable section of the human genome. By far. And especially in MHC3, the level of variation in that is 10 times greater than anywhere else in the human genome.
Where is that
MHC3. MHC3. The variation is 10 times greater than it is anywhere else in the human genome. And a lot of this has to do with copy number variants. Okay? Now, there aren't as many copy number variants in these regions as there are single nucleotide polymorphisms, but since copy number variants can deal with millions of bases at a time, the overall difference is significantly greater. Okay? And this is what makes it to where even your full siblings oftentimes are not a great match to donate an organ to you. Right? Because their MHC region can be completely different than yours. Blood type can be the same, and often is, but their MHC regions are different enough that it's not a good match. All right. Make sense? So how are we different? Well, it varies, but we're about 0.5% of our genome varies from person to person. About 0.5% of our genome.
So with the MHC region, how hard is it to find a matching organ donor?
It's pretty hard.
Like,
Yeah.
What are the statistics on that?
Well, it depends on which form of it you have. There are some variants that are more common than others, and so it depends a lot, you know. But it usually takes quite a time to find a good match when you're on a transplant list.
Is it similar to blood type where like people have like similar ones?
Except for, yes, but there's way more variation than blood type. Yes, yeah, because there are only 8 different blood types. I mean, normal blood types. There are like obscure blood types that have very rare sugars presented on the surface of red blood cells. But there are literally tens of thousands, if not hundreds of thousands, variants in these MHC regions. Yeah.
So like if you do find like a good match by looking at that region, like, but like they still reject it, like what happened? What went wrong?
I mean, it could be a lot of things. It could be something, you know, that's almost like a autoimmune issue, that it's just a slight, I mean, their genome's different, so their protein's still on the surface, just random protein channels might be different enough to trigger an immune response. Yeah. And that's why usually when you get an organ transplant, you are put on anti-rejection medications. But the interesting thing about this is keeping in mind that copy number variants change through the life of an individual. If you took stem cells from your umbilical cord at the time that you were born to save them for the chance that you might need to grow something from them, 45 years later, your cells are going to look different than they did at the time that you harvested those stem cells. So you're like, there could be a challenge even dealing with your own cells because your genome has changed throughout your life. Here's where the induced pluripotent cells come from, right? Basically take a cell that's lived your entire life and just trick it into thinking it can be anything. It can be anything it wants to be. All right. So we have a question that comes up that's a really important question to deal with. And that's this one. What is the humankind? So if we have almost undisputed evidence that our species, Homo sapiens, hybridized with Homo neanderthalensis and possibly Homo erectus as well, if these what the Denisovans were, Homo erectus, then it's like now we've got a really, really interesting picture going on. Okay? Where you have our species hybridizing with other species in our genes. Yeah, John.
What evidence is there for Neanderthal and Homo erectus?
That they existed, or what do you mean? That they interbred with our species?
That they existed.
Oh, that they existed? I mean, just being able to find a burial site where you have several specimens from that species. Which Homo erectus, until the Denisovans, this site in Europe was found, there was very little evidence of Homo erectus, but now if these are Homo erectus, there's a lot. But anyways, finding a site where you've got a number of specimens, and they have genetic and morphological distinctions that are make it very easy to take an individual and match it into that group.
Well, I thought Homo erectus, like Peking man, that kind of stuff, I thought that was largely, like, turned out to be false.
The Piltdown man did. It was a complete fabrication. But the other examples
that Peking man was as well.
No, it's not. No, it's legit. Yeah. The Piltdown man was a fabrication that was a bunch of different human skeletons mixed with ape skeletons. But no, I mean it's legit. And then with this site, the Denisovans, I mean we have overwhelming evidence of something that is morphologically, genetically distinct from us, enough so that you could classify it without any issue. Now, when you're talking about what that actually means, we'll talk about that in a minute. But Chad.
So that's how they go to these sites and say this is a different.
Yeah, it all started morphologically.
genetic.
Yes, it all started morphologically. That you could look at, say, a Neanderthal, and you could undoubtedly characterize it as Neanderthal and not Homo sapiens.
Because of the genome.
Because of, well, it started with morphology.
Okay.
But then when you look at the genome, you can do it genetically sometimes easier than you can the morphologically. Now, what that actually means, the difference between us and Neanderthals is anywhere, depending on your heritage. If you're from overwhelmingly European or overwhelmingly Asian descent, you probably only have about 1,000 amino acid differences between you and a typical Neanderthal genome. About 1,000 amino acid differences. Okay? Which compared to something like us and chimpanzees, you're talking about us and chimpanzees is more like, I have the number, but it's in the tens of thousands of amino acid differences. Okay? And so the amount of difference is significant enough that you could classify them genetically, but it's not huge, it's not insurmountable. And it's somewhat equivalent to the amount of variation present in our species now. So like if you compared like a very reproductively isolated group of humans to to a typical human genome, it'd be about the same amount of difference as a typical human genome and a Neanderthal
So
genome.
we still call
Then we still call it
isolated or
humans.
Homo sapiens.
Yeah. Yep. And so we can deal with it in the way of like we shouldn't classify them as separate species. That these should all just be a single species. That I don't think is as big of an issue on deciding what to do as being able to realize that the humankind is more diverse than the present diversity on Earth. That at times in human history that the amount of diversity was greater than it is now. And that shouldn't really come as a surprise. That shouldn't really come as a surprise. But it's interesting. So if you're from predominantly European or predominantly Asian or both, predominantly European and predominantly Asian, you're anywhere likely from 4 to 7% Neanderthal in terms of that your markers match better with the Neanderthal genome than they do from a genome of our species several thousand years ago. And if you are from oceanic descent, you are probably anywhere from 4 to 7% Neanderthal and 4 to 7% Denisovan as well.
Where's ocean?
So oceanic regions are going to be like Indonesia, some of the Polynesian islands, the very northern stretch of, the Aboriginal groups in Australia and New Zealand. Now, typically now when we think somebody from Australia or New Zealand, we think of the descendants of the criminals that were sent to Australia by the British Empire, right? They're very European. But I mean, they're like native people from Oceania. Yeah.
Why did we see the disappearance of these like variations?
It's a good question. I would say we haven't because their genome is in our genome, so they didn't disappear. What you see instead is a hybridization of a very rare morph of our species into the more common morph, the more dominant morph. That they haven't disappeared. They just we hybridized. Yeah.
From a creationist standpoint, how would you explain the existence of Neanderthals in this?
That they're all post-Adam. That all of even if you are going to leave it as separate species, which, I mean, again, you can say that there's really no reason to characterize them as separate species if they're interbreeding. I mean, by the biological species concept, they should be a single species. But regardless of what you want to do there, even if you leave them as separate species or unite them into one, that all of these morphs are post-Adam. Okay? And all of the variation is post-Adam. And then so then you have a question, well, what did Adam look more like, right? If you had Adam's genome, would it be more like an early version of our species or a modern version of our species, us, or would it be more like a Neanderthal genome? And one, I think that's irrelevant because they're all human. And two, I don't know. That's why it's nice to start with, I don't know if it's relevant, because then when I say I don't know, I don't feel so bad about not knowing. Yeah, Sadie.
Well, how would you explain the physical distinction?
Same way you would explain physical distinctions in really reproductively isolated groups of humans now.
So just the fact that they were just separated from each other and then...
Yeah, so if you got like the pygmy groups of Oceania, New Guinea, the Aboriginal people that just have a very different morphology than your typical member of our species. I mean, again, the amount of genetic variation among all living humans is not all that much different than the amount of variation between my genome and a Neanderthal genome. Just like the amount of variation morphologically among all living humans is not all that much different than the variation between my morphology and a typical Neanderthal morphology. That the amount of variation is fairly consistent. The difference is, if you take any modern human alive now, even from a really reproductively isolated group, you would probably be able to match them to our species versus, Neanderthal or Homo erectus, or some of the other members of our genus.
How would an evolutionist, because with other cases of reproductive isolation, reproductive isolation, the population that was once the same, because they have become isolated from one another, they can no longer interbreed and produce a viable offspring, which would categorize them as two different species. How do evolutionary biologists categorize, like, because of
If we're interbreeding with Neanderthals, that the isolation wasn't enough to create complete reproductive isolation. So the differences are the result in some genetic isolation. That's why you get a different morphology that's unique to particular regions.
But it didn't extend.
But it didn't extend to the point of generating reproductively isolated groups. Although the ability to do that would not be very difficult. What it would require is just maintaining geographic isolation. And that's hard in today, but it wasn't hard in human history to maintain reproductive isolation. All right, well, we're out of time. And we'll have to finish up these slides. Oh my gosh. Oh no, you're not going to be okay for the quiz. Not even close.